some more of our class. Alrighty. How are you guys? You guys are okay? All right, everybody's coming in. We got to get started here. All right, let me see those beautiful models finished and complete. All right, we'll go around.
Wait, Mr. Torres
Yes, Aaron.
was doing class?
Yeah,
Oh,
like right now. Like, show me what you got.
okay.
All right, Jaden, what do you have for us?
Okay, so this is probably the ugliest thing you've ever seen. Okay, I have the diagram, so I'll explain it.
Okay.
Okay. So,
Hey.
so these are the hydrophilic heads,
Okay.
and they're Q-tips. And then, obviously, these are the, what is it called? The hydrophobic tails.
Okay.
And then this one is, so I use pipe cleaners. So this one is supposed to be the, I think, alpha helix protein, something like that.
Yeah.
And then this one is a different type of protein. Let's just say it's a, a peripheral protein.
Okay.
And then there's one of these things here. And then there's two in the back. And these are the cholesterol.
Okay.
And then there's like a green thing here. And this green thing is supposed to be the glycolipid.
Nice. It looks pretty good. Okay, nice job. Cheyenne.
Mine looks like almost the same as Jaden's.
Okay.
Yeah, so this is my model. And then this is the protein channel. So it's like a straw, so it can go through. And then this is the alpha helix protein. It has like the little string thingy on the, and it's like twirly. And then these are, carbohydrate.
Okay.
Yeah. And these are the surface proteins. And then this is a globular protein. And this is the integral protein.
Okay.
And then this ball right here is the I can't, the glycolipid.
Okay.
The glycolipid. And then these Q-tips are the phospholipid molecules. And I think I explained everything.
Nice.
Oh, right. The over here, like the yellow things over here are carbohydrate. I mean, no, cholesterols.
Okay. Hey,
Yeah.
nicely done. Looks good. Great job. Aiden, do we have more additions?
Yes, I had more, but the Q-tips started to fail me because I wasn't allowed to waste any more Q-tips.
Okay.
So, yeah, we'll try and not break it here. I bring the masterpiece into view.
Okay.
So everything's still the same, but added is a Torx bit, which is a peripheral protein, and it's magnetic. So I took this like keyboard stand thing I have to prop a keyboard up and attached it to that, and so that's supposed to be like a surface protein. And then I also took the cap from my pen and added it in, and this is an integral protein that because it's like longer, and yeah, those are the additions I was able to make to it.
Nice.
Additions I was able to make to it.
Nice. Mindy.
Mine's way bigger than others. I put it on like this little stand.
Okay.
You like it?
Nice.
So this is the hydrophilic head, and this tip, and then this is the hydrophilic tail, and these strings right here is the alpha helix protein, and this yellow is the you were explaining yesterday as the fur jacket, the yellow fur jacket, the cholesterol. Yeah. Yeah, that's all I
All right. Ray.
Okay. So yeah.
Hey, there it is. Okay.
Yeah, so I like copied it off from like this one diagram, but I added like these blue marker, but you can't really see it, but that would be like the cholesterol, I guess, but that's one like big thing I just took off from like a paper.
Okay.
And that the blue things are like the glycoprotein.
Okay.
And then that's the glycolipid. Yeah, I couldn't add much to it anymore because like my hot glue gun kind of like didn't work, but yeah.
Yeah, it looks good. Nice job. Sharon.
So I'm like halfway done, but like there's like you can kind of oh my god, it's such a mess. So you can't really see, but yeah, I tried to like sew it.
Okay. Hey. That's pretty cool.
So I tried to make like French knots for the hydrophilic heads and then like the thread in between for like the hydrophobic tails.
Okay.
And then that purple palm is like, it can be integral protein or protein channel, I guess, like
Okay.
both work. And then the red is, it can be like a glycoprotein or a globular protein, also both work. And then I don't know if you can see, but like these beads, they're
Okay.
like the glycolipid, is that how you pronounce it?
Yeah.
And yeah, there's like still some black thread, so I guess you can call that the cholesterol.
All right.
It's a mess. Yeah.
Coming along. Aaron, do you have something to share with us? Aaron might not be there. All right. Hey, well, nicely done. They all look different, but overall, they are all achieving the goal. Okay. Remember that the goal is not necessarily to have the most beautiful looking membrane. It's awesome when you like the way it looks, but the goal at the end of the day is, does it actually interpret the structure of the membrane, and is it helpful to you in terms of identifying structures? That's really the goal. If it makes sense to you, if you can explain what pieces are doing what, hey, you've accomplished the goal. Now, please do not destroy them yet, as you are going to use them for your assessment on Thursday. Okay? You will be using yours on Thursday to complete your assessment. Today we're going to continue talking about membranes, but we are going to actually move over to not just the structure, but really what are they doing? Like, what's the function here? So we're going to look into transport. Okay? We'll do part of the transport today, another part of the transport on Wednesday, and then on Thursday, you guys are going to have an assessment, but it's not going to be a paper assessment. Okay? I will tell you right now roughly what you will be doing. You are pretty much going to use your models and you are going to create a lesson for someone using that model where you explain certain things. Okay? I will not tell you what you are explaining just yet, but when your assessment day comes, which is Thursday, you're going to use that model as a basis to create a short instructional video in which you pretty much create a lesson for somebody on a specific topic. Okay? I think that is so much more valuable to you in terms of learning the material, and you'll always be able to go back to that video when you are studying and use that as study material as well. Okay? So that is what your assessment will be. I will grade it based on, hey, are you actually answering the questions? Are you actually providing the right information? And I will give you feedback on your videos if things are missing. But for the most part, if you answer the questions that I'm asking you, you'll be good. Yes, Mindy?
You know how I didn't put every part of those other like globular protein or glycoprotein, like if I didn't put those, should I put that on my model so it could be better for when I make the video?
You should have a certain, like one of each type of protein on there. If not, you should be able to at least reference where that protein should go in your model. So think about that. Otherwise, you just need to be able to reference to it. Okay? All right. Notes for your lesson today are posted on RenWeb. What am I saying? On Neo. Sorry, I'm still on RenWeb. Along with that, you guys also have some guiding questions to look at. We are only going through passive transport. The questions that I provided for you on Neo are both for passive and for active. So tonight you will only work on passive, tomorrow you'll work on active. Okay? So let me go ahead and share my screen, and let's go ahead and do a little bit of transport here. All right, here we go. Yesterday we started talking about membrane structure. You guys created some very nice looking models, very much appreciated. You guys all did a great job. Very different in the way you approached it, but you guys all accomplished what you were trying to do. So now we're going to look into transport. Like, how does this membrane even do anything with that structure that I've been looking at, right? So we have active and passive transport. I've said this before, scientists are not very creative. So usually what it sounds like is what it's doing. So active is going to be like requiring energy, while passive, no energy. So if you need help remembering that, when you are active, right, when you're working out, you need energy. If you're just passively kind of hanging out, you can just kind of sit there. You don't need too much energy, okay? So when we talk about the phospholipid bilayer, which is what you guys have created different models of, we know that the bilayer is semi-permeable, okay? Meaning it's selectively permeable in a way that it allows certain molecules in and not others, okay? So selectively pretty much means, hey, I'm going to pick and choose, while permeable means that it allows entry and exit of molecules, okay? Some molecules can pass easily through what we call diffusion. Diffusion just means things just kind of enter, okay? Things that can diffuse through membranes are usually pretty small. They don't require energy, they can just go through. But if they're a little bigger, then we say that they go through facilitated diffusion, which is usually going to be a tunnel or a channel protein, okay, which will allow things to go through. Now, if you need energy, though, then we're going to be talking about active transport, okay? Large molecules are going to usually use active transport because they cannot get through by themselves, okay? So So small molecules usually can get through with very little energy expenditure. Large molecules usually need energy in order to get them in or out of the cell. Okay. The process usually to get large molecules in and out, we call it endocytosis or exocytosis. Endocytosis is entering, exocytosis is exiting. Okay. So far, so good? Yeah? All right. One thing I will point out here, polar heads, they attract other polar molecules, and then nonpolar tails, so the inside here, they will actually prevent passage of ions through the membrane. So charged molecules don't do very well if they are going to go through the phospholipid bilayer. However, you can get charged molecules to go through if there is a specific channel for them. Okay? But they don't just diffuse through very easily. Now, when we talk about diffusion, diffusion's passive, right? It is the passive movement of molecules from high concentration over to low concentration. Okay, that is key. It is key to know that in order for diffusion to happen, this passive transport, you go from a high concentration over to a low concentration. Okay? So you have a lot on the outside, it's going to slowly move into the inside to try and balance. Okay? So passive, no energy required, but the net movement is going to be the overall movement of the particles. Okay? So if you have a large concentration in one place, it's going to move until it reaches what we call equilibrium. They always want to move towards a lower concentration. Okay? So high to low is always going to be passive. Good? The concentration gradient is another vocab word for you. This is pretty much the difference in concentration of the substance in the two locations. Okay? So if you guys look at the picture down there, I don't know why my mouse isn't showing, but if you guys look at the picture that says try tutorials here, you can see that you have a concentration of IKI that's larger on the bag side than in the beaker. So that is potassium iodide, and that is going to cause the sodium iodide to go into the beaker because there is no concentration there. So it wants to balance out the concentration. Okay? Good? So here's a better picture here. If you have a bunch of molecules, as you can see in the picture on the left, there's a lot more on the left than there is in the right in the first picture. So then you're going to move the molecules over to the right. Now in the other picture, the picture that's on the right, you'll notice that it's pretty even. So you're going to have a very slow diffusion to try and even it out. At the beginning, it's going to go to the left, and then it'll go back to the right, so on and so forth. Does that make sense? So we always go from high to low. High to low. Certain factors that are going to affect how diffusion actually works. Number one, the concentration gradient, which is, hey, is there a big difference between the inside and the outside? If there is, then it's always going to go from high to low. The second thing that's going to affect the rate at which diffusion occurs is going to be surface area. Okay, if you have a large surface area, then you're able to move things quicker than if you have a small surface area. Okay, based on these pictures, left or right, which one has a larger surface area? What do you guys think? Somebody just turn on your mic and let me know.
Left.
Left, right? The left side has a larger surface area? Yes or no? Anybody?
Yes.
Are we talking about the circle?
Yes.
Or are we talking about that squiggly thing?
Circle.
Circle? You guys sure?
Yeah.
Is there a difference?
Maybe not completely.
I think
I don't know. It's really close.
it's pretty even, but the point is larger surface area will give you guys the larger diffusion rate. This is actually a pretty bad representation because a circle itself is actually pretty high in surface area. Would have been easier if it was like a triangle or something. And then the length of the diffusion path. So if you guys look here, a shorter path will enable a faster rate versus a longer path. Okay. So here are our actual answers. So if you guys look at the paths, so in terms of surface area, that star shape or squiggly shape should actually have a higher surface area. I can see why you would go with the circle as well, but because it's so like bent or folded, all of those folds should provide a larger surface area, thus creating the higher diffusion rate. And then at the bottom, we see the length of the diffusion path. If you have a short path, it's faster. If you have a long path, it's slower. Okay? Any confusion as to why the answers are what they are? No? You guys are good with that? I do get why some of you thought the circle. But when you fold something, anytime you want to increase surface area for anything, you fold it. Anything like that's super absorbent, for the most part, has a lot of folds in it. It allows for the absorption of things, even if you don't see the folds yourself. Whoops. So why is this important? Well, we are interested in being able to maximize surface area. The body does this in a lot of different ways. Different places that you can see this is in the alveoli and the lungs. Why would it be important to have a large surface area there? Anybody? If you think about your lungs, go ahead, Aiden.
Get as much oxygen as you can.
Yeah. So you want as much oxygen as you possibly can obtain. Therefore, the alveoli will help you uptake more oxygen than if there was no folds there. What about the cristae? The cristae are part of mitochondria. If you remember what the mitochondria do, why would you want a lot of folds there? Ray, what do you think? Why would you want a lot of folds in the mitochondria?
I honestly don't know, but I don't know, maybe for more space, like you mentioned.
But what does the mitochondria do?
Isn't that just like the power thing?
Yeah, you power things. So if you have a lot of folds, then you hopefully could actually harvest more energy. All right, and then the last one that root hairs for water and mineral ion uptake. So that one's a good one. But then villi, we don't have a picture of it. They look pretty similar to root hairs. They're just kind of little finger-like things. But villi are also in a structure that increases surface area, and that is for you to be able to uptake as many nutrients as possible from your food. Okay, so surface area, for the most part, always a plus. This is why cells remain small, and this is why things create large folds. Okay. Last thing when we're talking about passive for today, pretty familiar word to most is osmosis. If you watch the movie Osmosis Jones, that's kind of relatively about this, but osmosis really talks about water, right? The movement of water. And it's crucial that water goes into cells and out of cells because we're composed of a large amount of water. Okay, so when you submerge a cell into water, water molecules are going to pass through the cell, and they will go from low solute concentration to one of high solute concentration. Okay, so does that confuse you? Is there a confusion there with that statement? So you're going to go from an area of low solute over to an area of high solute. Somebody tell me what solute is? Solute usually goes with the word solvent. So what is solute? Anybody know what solute is? Is it the thing you dissolve into the solution, or is it the thing that is dissolving? Mindy,
We dissolve it in?
the thing you dissolve it in?
Or no, I meant the other way around. I meant
The thing you're dissolving?
being.
So solute refers to what you are dissolving into a solvent. Okay? So when it says that it's going from an area of low solute concentration to an area of high solute concentration, what is the water actually doing? So if you think about that statement, you're going from a low solute concentration to a high solute concentration. What is the water doing? Is it increasing the concentration or is it decreasing the concentration of solute? What do you think, Aiden? So if you're going from low solute to high solute, solute is the thing that is dissolved into the solvent.
Mhm.
Is water causing the solute concentration to come down or to go up?
Go up.
It's actually going to cause it to go down, right? What you're doing is you're actually causing a dilution. So water is going into the high solute concentration, thus making the concentration overall lower, okay? So here you guys can kind of look at this. You have three different types of osmotic control, and this is very crucial. So if you have a hypertonic solution, okay, you're going to have water coming out, okay? So water comes out of a hypertonic solution, and that causes the cell to shrink, okay? So if water comes out in a hypertonic solution, that means that the outside is very high or low concentration, high or low concentration. If water is going out, is the outside high or low?
Hi.
High, right? It moves towards the high. So in a hypertonic solution, it means, hey, the solute concentration is higher on the outside than it is on the inside, so water is going to come out. On an isotonic solution, it basically means that the entering and exiting rate is equal. Nothing happens to the cell. It means that the solute concentration inside and outside is the same. It doesn't mean that it doesn't do anything. It just means that the amount going in and out remains the same at equilibrium. Therefore, you don't observe a change. Now, hypotonic, right, is going to cause water to flood into the cell. If water floods into the cell, it means that the solute concentration is higher or lower inside. Is it higher or lower on the inside?
Higher in the inside.
Higher on the inside, right? Because we always move from low to high concentration. So as the water rushes into the cell in a hypotonic solution, it causes the cells to plump up or get really fat. We call it hypotonic. So if something's hypotonic, it always gets bigger. If you want to remember it as a hippo, right? That works. So hypotonic means the cell gets bigger, it gets filled. Oftentimes, if you put cells into a hypotonic solution, many of the cells will actually burst because they cannot handle the pressure that's built by all the water rushing in. Ideally, you would want your cells in isotonic solution because you don't want them to shrink and you don't want them to get really pretty much pressurized and flooded. Okay? All right. I will not have you do analogies today. So, some key things that I need you to really kind of put your attention towards. Number one, when we talk about osmosis, okay, or water movement, we got to remember that it goes from low solute, okay, that's key that you remember, low solute concentration to high solute concentration, okay? That is the biggest key statement that you could write if you are thinking about osmosis, okay? When you're thinking about osmosis, it goes from low solute to high solute, and we have three different things that can happen osmotically. Those three different things are going to be, a solution could be hypertonic, isotonic, or hypotonic. In a hypertonic solution, the cells shrink. In an isotonic, the cells remain the same. In a hypotonic, the cells plump up and get bigger. Okay? Osmosis and passive transport or diffusion, they are both passive. They do not require energy. Passive transport, for the most part, is going to require just being small and not charged. Questions? Please ask questions. It is a lot of info. Yes, Mindy.
Are you gonna upload the slides today?
They should be up already. 1.4 under resources. It says membrane transport. Should be under 1.4. Huh?
The passive one or active one? Because they're both the same.
Passive. We only talked about passive today. Yes, Cheyenne.
Oh, so osmosis is passive?
Osmosis is also passive. However, we only use that term when we are referring to water. And one thing that I didn't actually talk about is aquaporin. Aquaporin is actually a protein that's going to facilitate water molecules moving in and out. Okay. There's not much more that you need to know about aquaporin, except that it's an integral protein. And that integral protein allows for water to move in and out easily. It's basically the way water can get in and out, even though water is technically polar. Okay. Any other questions? I am more than happy to answer them. Please don't keep them to yourself. Yes, Mindy.
Teacher, the assessment you're talking about the beginning of class is Thursday, right?
Your test?
The assessment, yeah, the video assessment?
Yes, that is on Thursday. Yes, Aaron.
Sure. When can I present my thing?
Tomorrow.
Tomorrow? Okay, thank you. All right, then.
All right, any other questions? Questions, concerns? I feel like we're walking with Aaron somewhere. All right, so on NEO, you guys have a very short assignment simply to really kind of go over what we talked about today. You're only working on the passive transport, so you have a few vocabulary words, some short answers to do, some structures, and that should be it. Okay. You don't have anything to build today, so that's a plus. And you don't have workbook pages just yet. Please make sure that your workbooks come in at some point within like the next 2-3 weeks if you don't have your workbook yet. Chapter 1 is still posted for you guys to access digitally. If you don't know what I'm talking about in terms of the workbook, you should have a workbook that has an owl on it. It says IB Biology Workbook. If you don't know what I'm talking about, email me and I'll send you the link. All right. All right, we have about 15 minutes. I'll let you guys work on those questions for now. If you have questions on those, please let me know. If you don't have questions, I think you can get them done. Yes, Mindy.
You know the passive transfer sheet, should we print it? And
You do not need to print it.
Rather,
You can just type it if you want, and then you can submit it into the Dropbox that I'll put up right now.
that's due today, right?
That is going to be due by tomorrow, but yeah, submit it by today. All right, there is a Dropbox for your responses that is there for you. I don't think Neo's playing games with me today. I think it's there. All right. Let me know if you have further questions. I'll be right here.
Mr. Torres,
Yeah.
is there a way that you could upload the file of the worksheet so that I could download it?
It doesn't let you take it. It doesn't let you print it.
No.
Not even if you right click on it.
Um,
Oh, never mind. I tried that. It doesn't.
yeah.
Um, add file. Um, I need to get it from my drive.
Sorry.
Wait, Mr. Torres,
Yeah.
we do all the questions.
Just the passive.
Oh, okay. Thank you.
Da-da-da-da. Okay, so here comes that. Add. File. Download. All right. The worksheet is uploaded as a doc for those of you that want it.
Thank you.
They're on resources. Da-da-da, da-da-da.
Mr. Torres, can I go use the restroom?
Go for it.
vitamin D.
So were we supposed to just email you the assignment?
There's a Dropbox.
Believe so, even though I thought there was a Dropbox. Let me just
All right, you guys have about 2 more minutes. 2 minutes.
Oh, sorry.
All right, 1 minute, guys. 1 minute. Just kind of finish up that.